It's time for diagnostic microbiology and immunology. This chapter has 3 parts. First part, healthcare related infections. Second part, microbiology and immunological testing. Third part, vaccinations. Let's start with healthcare-associated infections, abbreviated sometimes HAI with an S at the end. The other term you see for it is nosocomial disease or nosocomial infection. Nosocomial relates to the word hospital. However, do not just think that these are infections obtained by patients in the hospital. You could be a healthcare worker exposed to the coronavirus that is ever present now and pick up a nosocomial infection from a patient. And it does not even have to be in a hospital. It could be dental, office related, home health care, ambulatory clinic, nursing home. So I'll let you look at these statistics here and let me go on. This is a study looking at hundreds of HAIs, and I just wanted to point out the top 3, actually top 4 here. Pneumonia, surgical site infections, GI tract infections, and UTIs. Who are the high-risk populations? Many, for different reasons. Those who are immunosuppressed or immunodeficient have lagging immune responses. The elderly, because of decreased immune competency. People that are having surgery where their first line of defense, the skin, is breached. People who are having instrumentation put into their body via maybe, let's say, a catheter or something put into a vein, maybe a trach tube, some instrumentation that bypasses their normal defenses. Any of these are ways that microorganisms can get into the body. So you can see that these high-risk populations relate to the top 4 infections that I just laid out for you on that chart. Here is another way of looking at the top 5 infections, but from the standpoint of the microorganisms that cause it. So I put red boxes around the top causes of each type of infection. You'll see that some of these organisms are found in multiple categories like Staphylococcus aureus, Klebsiella pneumonia, E. coli, Enterococcus. You'll also see that there are some microorganisms that are particular for a type of infection. So for GI tract infections, particularly colitis, we have Clostridium difficile, which is a normal flora organism in the colon. Over at the top, at the right, you see that column labeled bloodstream infections. And I wanted to point out one of the only non-bacterial organisms here, Candida species, as it's listed, but it's probably going to be Candida albicans, the most common pathogen in that genus, is the number 1 cause of bloodstream infections. And remember, since it is a fungus, it's going to respond a different way to chemicals, so you cannot give it a typical antibiotic like you would a bacterial infection.
Wait. That's not possible.
So here's a look at types of procedures on the right-hand side that could predispose you to infections if you were having any of these things done. And on the left table, you see transmission methods. Let's change gears here. Now we're going to move to work done in a laboratory, and much of this has already been covered in lab, so you don't have to be going over this again. You've already seen and done microscopy in the lab. You have grown microorganisms in the lab. We have done biochemical testing, and you have seen a rapid technique like an API 20. You've identified bacteria in the lab already. You've done staining, wet mounts. You've grown bacteria on different kinds of differential and selective media. Typically you do not do tissue culture in a microbiology lab, in a virology lab maybe, or some other types of courses. And there are all sorts of identification tests that can be done that we have not even talked about. One of the things done very often in a lab is a simple rapid technique for identification called the API 20. You see it in the top right of this picture. There are two APIs there. They're called API 20 because the 20 signifies 20 tests that are run. API just stands for the company that used to be that made this originally, Analytical Products Inc. These are test strips. They have dehydrated media in them. You hydrate them with your bacterial suspension, incubate it. Occasionally you have to add a reagent. You look at the color changes. You compare that to a chart of colors. You determine which are positive and negative. And then you put the positive and negative test results into a computer which searches its database and spits out the name of the organism for you. Taking this one step further, there are some more sophisticated machinery that you see on this page also, of different types. Hospital may use one or another, or sometimes they may use a couple of them. And they are now the automated identification procedures done in labs. These are very expensive, very small labs that may only have a couple of dozen identifications of bacterial infections every day, may not be able to pay for these sophisticated types of automated identification machines. Or maybe they might use the API for identification or some other rapid technique. By the way, the company makes other test strips for other types of microorganisms. Or some small hospitals may send their lab work done for identifications out to a reference lab. And of course, you are familiar with identification of a bacterial unknown. So here would be one of the tests that would typically be done. This is a test for antibiotic susceptibility on a bacterium. These are two pictures that illustrate what is called the MIC, which stands for Minimal Inhibitory Concentration, also called the dilution test. So let me take you through this. So let's start on that picture up at the top left. And what you are looking at is a dilution of one particular antibiotic, antibiotic X. And over at the far left, it is in high concentration at 32 milligrams per liter. And we are doing twofold dilutions, so going from tube 1 to tube 2 you notice that the concentration goes from 32 to 16, and then we do another twofold dilution, 1 to 2 dilution, and now we've dropped it by another half, and therefore you can see that as we go to the right, the dilution of the antibiotic is becoming greater, and the amount of the antibiotic in the solution is decreasing. And then we've got a control and another control with no drug. Now, we are going to inoculate each of these tubes with a bacterium. Let's say that Joe has a staph infection. We've already done the identification. We know that it's Staphylococcus aureus, or maybe we don't know the name of the organism yet. It could be that we're running the identification at the same time that we're doing this antibiotic sensitivity. So we now take a culture of that microorganism from Joe, and remember, you are looking at the dilution of one drug only, and typically you would be doing this on a couple of dozen different antibiotics and synthetic agents, not just one antibiotic. In any case, you add the same amount of that Staphylococcus to each tube, except the sterility control that's second to from the right. And you will notice that there is no growth on the first 4 tubes, and then the odd little pattern of black and white indicates growth of the bacteria, and you notice that it grows in the last 4 tubes. So in those 4 tubes, the dilution of the antibiotic has become so great, and the concentration of the antibiotic is so low, that there is not enough to inhibit the bacterium. So the circled tube is the highest dilution of the drug, or the lowest concentration of the drug, that still inhibits the bacterium, and that is the MIC. Now, at the bottom, you see a what is called a microtiter plate, and this is about a 4 by 6 plate, 4 by 6 inches. It's got tiny little tubes that are maybe half an inch in height and maybe a 1/4 inch across. Each one of those circles stands for one of those little mini tubes, and you can run the same dilution that I took you through on the tubes above in these microtiter plates. So we have 10 tubes in that picture at the top. Notice we have 12 of these little tubes in the microtiter plate labeled 1 through 12, and we've got a control. So again, we start out in the little tube 1 with the highest concentration of the antibiotic. Tube 2 to the right will have half, tube 3 half of that, 4 all the way through 11. Now notice the rows. We could run 8 different antibiotics on this microtiter plate. We have 8 rows. This is done generally automatically with sophisticated pipetters these days, and you can put the whole microtiter plate on a reader that will identify the growth. So if you look at row 1, there is no growth from tube 1 all the way to tube 8, and then the yellow indicates growth of the bacteria. So again, the last tube that has inhibited growth is called the MIC. This is a typical test that is run all over the place to do antibiotic sensitivities. Now, the way you may have done it in lab, the way we did it in lab, is called the disc diffusion test, or the Kirby-Bauer, named for a couple of people that wrote it up. It's been done for a very long time. It's typically done in a laboratory in a microbiology class because it's easy to do and it's not as cumbersome as all the dilutions required for the MIC. In this procedure, the microorganism in question has been streaked on a plate. You can see the opaque streak lines of the bacterial growth in the background. Then, in this case, about 8 different antibiotic discs have been placed on the plate. The antibiotic has already saturated the disc, so you actually buy the disc with the antibiotic already on it. We have 8 different antibiotics on these discs, and they are labeled. And the plate is incubated after the discs are put on it. So again, you swab the plate with the bacterium, put the antibiotic disc on top, incubate it, then read it. And you look for these zones of inhibition, measure them, look them up on a chart, and the chart will tell you whether the organism is sensitive, resistant, or intermediate to that particular antibiotic. You can't just look at this plate and say antibiotic 3 is a good drug. Even though there is a small zone, based on the chart data, it's not a large enough zone to make that particular antibiotic 3 a good one to choose if you're trying to get rid of a person's infection. Certainly drug 6 looks a lot better. The larger the zone, the better the kill. Now, the advantage of the MIC is because you are doing dilutions and you know concentration of the drug, it gives you a much more fine-tuned idea of dosage, whereas the Kirby-Bauer does not. You buy the disc from a supply company. The disc has only one dose of the antibiotic on it, and that's all you know. Now, the reason I brought that up is that there now is a newer type of antibiotic testing that some labs do, which is kind of a combination of the MIC and the Kirby-Bauer. And it's the plate that you see in brown. You can see the growth of the bacteria that's been swabbed on the plate. And you see a piece of paper here with something that looks like an E. This is called an E test, by the way. But right below that E letter is TX, which might be tetracycline, let's say. So this disc has been saturated in varying amounts of tetracycline. Up at the top, you see 32, highest concentration. Down at the bottom, you see 0.002, most dilute. You see a zone of inhibition up towards the top, and where the edge of the zone crosses the saturated piece of paper, whatever that reading is your piece of data. In this case, it would be 1.5. So this represents concentration of the drug. As I said, an interesting combination of some of the facets of the Kirby-Bauer with some of the facets of the MIC, and other looks at Kirby-Bauers with 3 different organisms. So let's go to immunology and what types of tests may be done there. When would you do testing in an immunology lab on a patient's specimen? Well, if the organism that is causing the infection cannot be identified easily in a laboratory, in a typical bacteriology laboratory, so maybe the infectious agent is not bacterial, it's viral, and there's no other easy way to identify the virus other than by doing an immunological test. Or maybe it's cheaper to do this test than it would be to run a variety of biochemical tests on it, the typical way. So we are now in the realm called serology. Serology is the study of serum. So what is it used for? You can use serological testing, and these are immunological tests that we're going to be talking about, for a couple of different reasons. You can use serological testing to test for the presence of antibody in the patient's blood to a particular infectious agent. Maybe you want to look for antibodies to HIV in the patient. You're testing for the antibody. Now you have to use a known antigen. a protein of HIV to find that antibody, but you can buy that. Let's turn it around. Maybe you want to look for the antigen itself. You want to look for the infectious agent in the patient, not the antibody. So in that case, we're going to look for the antigen, and we're going to use a bought antibody to find the antigen in the patient. So you can use serological testing both ways. Another thing that serology tells you is if you are looking for antibody and the patient has had this particular infection and they have antibody to this agent, like HIV, you might want to know how much antibody. What is the level of antibody? If the patient is in the throes of the infection or getting over the infection, even more so, they're going to have a high antibody response. If, on the other hand, they had this infection 2 years ago, and this is not even the infectious agent that they have, you may think it is, but it's really not. It's a different infectious agent. But let's say that they've had this disease already, but it's been 2 years ago. Their antibody level is going to be lower. They will still have antibody, but it will be a much lower amount. So antibody testing gives you some quantitative information about, does the person have this particular disease? Do they have it now? Did they have it before? And it might even tell you where they are in the disease itself. So when we're talking about amounts of antibody, the term for that is called titer, antibody titer. And you're looking for antibody in serum, hence the word serology. Serum is basically a variation of plasma. It is the fluid of the blood, but it has no clotting factors there. And you have highly concentrated antibody left in there. So let me take you through this. Let's say we have a patient here, and maybe they have a respiratory infection of some sort, but it could be the flu, and it could be any number of other viral types of agents. Let's say we've eliminated all the bacterial possibilities because we've tried culturing it and it doesn't grow any bacteria, so we think it's viral. But it could also be the virus that causes COVID-19, right? Could be the SARS-CoV-2 virus. Or it could be some other virus that causes a respiratory problem. So we need to look for specific antibody to the possibilities. Maybe one possibility is a flu strain X. Another one might be flu strain Y. Another possibility might be the coronavirus that's causing COVID-19, or maybe it's another virus. We'll call it Z. All right, we're going to look for antibody against these different viruses in our patient. So, in the picture, over at the left, starting out, looking across left to right, you'll notice that we are making dilutions of the patient's serum. So, highest concentration of antibody at the left, 1/20. Most diluted, lowest concentration of antibody at the right, second from the right, 1/640. And usually it would be taken out more than that, 1/1280, 1/2560, and so on. Now, we've got the dilutions of the patient's serum. We're going to add a particular possible virus. Let's go with the coronavirus. And notice it incubated. Down here at the bottom, you see the results. So we have some sort of agglutination or reaction, visible reaction going on in tube 1, in tube 2, 3, 4. But when we get to tube 5, again moving to the right, there is no visible reaction. 5, 6, and 7 all have no reaction. So the last dilution of the serum, which still gives a visible reaction, is our titer. And if you look straight up above at the tubes amount of dilution, you're looking at a 1 over 160. That would be the titer, 1 over 160. Now remember, that was to the coronavirus. Let's say that we also have run the exact same test with flu strain X. And on that test, when we ran flu strain X, we get a titer of 1 over 1280. Well, 1 over 1280 is more highly diluted than in the first case, which was against the coronavirus. So the person must have flu strain X because they have more antibody. Again, we could dilute out the patient's antibody further, 1 over 1280, as opposed to 1 over 640 in the case of the coronavirus, and we still get a reaction. So the person does not have the coronavirus, they have a flu strain X. And again, this is done in an automated way now. By the way, most often they take the numerator 1 over and kind of throw it away, and they just use the denominator number. So in reality, you would say the titer is 160 or 320. Now, as we move into the testing, you'll notice that we are going to be using some known antibodies. If we're looking for an antigen, like maybe an HIV antigen, in the patient, we're looking for the HIV virus, we have to use a known antibody to detect that antigen. So the question is, where do you get those antibodies? Well, they most often make them in the form of monoclonal antibody, or sometimes you see them written as MAB. These are antibodies that are obtained using animals. I don't really want to get into the mechanics of this. I think you can follow this on the picture. We produce hybridomas, which are combinations of tumor cells and activated B cells that make a particular antibody. And the result of that is we get very pure antibody to a particular antigen. So those little Y-shaped molecules, which are antibodies, might be antibodies to HIV, and then we can use those antibodies in a test kit to find the HIV antigens that we are looking for. These MABs can also be used in therapy to give to patients. They're very often used in treating cancer and a whole variety of different things. So let's get going in the tests. One of the most common tests done, you might have done this before. We typically do it in a lab. It's called an agglutination test. Let me have you look down at the blood card down at the bottom. Blood typing uses this procedure. So to remind you, when you're doing blood typing and you're looking for those A antigens on the red blood cells, or the B antigens, or the Rh antigens on the red blood cells, correct? You use antibody to detect the antigens. Remember, there were 3 little bottles of fluid that you added to the drop of blood and mixed them up. One of the bottles was labeled anti-A, one labeled anti-B, one labeled anti-Rh. So on that card, the person's blood has been put on the card in 3 different places, and each of the 3 antibodies has been added, anti-A, anti-B, anti-Rh, and we are looking for clumping or agglutination of the red cells as they pull into visible clumps. And you can see that in the first well and in the third well, and that means that these red blood cells have got A antigens on them, and the well to the right where it says anti-D, that's a major Rh antigen. So the person has the Rh antigen on their blood cells, but not the B, because you see no agglutination in the B middle well area. So this blood is A positive. Agglutination testing is done in a variety of different forms. Very often there's a common Staphylococcus aureus agglutination test. There is a agglutination test for syphilis that employs this procedure. The monospot test for infectious mono, called the monospot, uses this procedure also. Fluorescent antibody testing. Pretty simple to do. You can do it, again, you can be looking for the antibody or you can be looking for the antigen. Let me point out on the picture here, you have a fluorescent microscopy view. This is a neuron in the middle, and those bright green spots are rabies viruses that have become fluoresced with the use of a fluorescein dye attached to an antibody. So this is probably a brain cell that we're looking at here, because they typically do this on brain tissue, and they've used a known antibody to the rabies virus that is tagged to a fluorescein dye, and when it finds the antigen, it attaches to it and it fluoresces. That's basically the procedure in a nutshell. And there's a direct procedure and an indirect procedure. So many viral infections employ the fluorescent antibody test procedure. The enzyme immunoassay. Sometimes you hear it called the ELISA for enzyme. I've just forgotten what it stands for. Immunosorbent assay. Enzyme-linked immunosorbent assay. Most of the time it's just called EIA for enzyme immunoassay. Good view of one of those microtiter plates that I was talking about earlier, and you can see the pipettes there. So here, an example would be HIV testing for antibody. Let me take you through the procedure real quickly. These are the little wells that you see on that microtiter plate. So we have, let's say, an HIV protein, a known protein, bought, that is represented by that blue little triangle right there. And we are going to coat the little wells with it. Then we're going to add patient's serum. You can see that black antibody molecule right there. Then we wash off the antibody that's not attached to antigen that's just floating around, washes away, but if the patient has antibody to the HIV, it attaches to the protein right there, as you see. Then we add another antibody, a second antibody, and this antibody is attached to a detector. The detector is this red object here. Now this antibody is antibody to the person's antibody. That is, this is antibody made against antibody to HIV. It's antibody made against antibody. I know that sounds strange, but it is. So for example, we could take antibody made by people against HIV, if they're infected with HIV, put it into a horse. The horse would make antibody against the antibody protein antigens we just put into the horse. Antibody against antibody. Now, if the patient has HIV antibody, remember the first black antibody Y-shaped molecule, and we've added a known antibody to that antibody, to the well, then they will react, attach to each other, and that causes a color change in this detector chemical that's in red. In this case, the well turns blue. And again, you can see this visibly, or you can use a machine to read it. If the patient does not have antibody to HIV, they have not been exposed, then there would not be this first antibody. Without that first antibody, when you add the second antibody, there's nothing for the second antibody to attach to because it doesn't attach to the antigen. The second antibody only attaches to the first antibody. Therefore, you get no color change. Here are some uses for the ELISA. So again, ELISA stands for Enzyme-Linked Immunosorbent Assay, but usually now just called Enzyme Immunoassay, EIA. One of the most common types of tests. The immunoblot, the Western blot test. We have talked about the Southern blot already, and this is a variation of it. This is a better test if you're testing for HIV disease. It is a more definitive test, whereas you can get more false negatives and false positives with the ELISA for HIV. So the procedure. First of all, we are going to use HIV antigen. So we break up the HIV antigen. It's a known antigen. And we do an electrophoresis on it. So you see electrophoresis here in the bottom left picture. And we have a breakup of the proteins of HIV that have migrated during electrophoresis from top to bottom. Now we're going to transfer these protein pieces from the gel to a filter paper, in this case made of cellulose. That's why this is called a blot, and it can be done in different ways. I'm not going to get into how they make the blot. Then, after it's blotted onto the paper, and by the way, the reason for blotting it onto the paper is that the gel is hard to handle. It breaks apart. So to stabilize these protein fragments, it has to be transferred to the filter paper. In any case, now we are going to use patient's serum if the person has antibody to HIV. That antibody will attach to these protein fragments on the paper. We can use another antibody to attach to those antibodies, or we can use some sort of other detector system to detect the antigen that has attached to the antibody. Maybe we use some radioisotope, something else, and that will give us a definitive identification of the presence of antibody in the patient. Another look at the Western blot. Now you can see better how the blotting is done to transfer from the gel to paper. Here's another procedure. I did not even have this in my PowerPoint, but I wanted to add this because we do this procedure in lab, and this is a really quick growing type of assay. In fact, the new procedure that is going to be done for antibody testing for the novel coronavirus is going to be done by this way. You also see an example of a HIV test on the far right. This test is also done all the time in vet clinics on heartworms for dogs, strep testing for strep throat in children. Pregnancy test kits are based on the same procedure. In that case, they're looking for a hormone called human chorionic gonadotropin, which indicates pregnancy. So you can use these little objects that we'll talk about here in more detail to find the antigen in the body. For example, let's say you're trying to identify whether the child has strep throat, so you're looking for streptococcal antigens in the throat. Or, in the case of the OraQuick at the far right, this is used to identify the antibody to HIV. So again, you can be looking for antibody, you can be looking for antigen. Okay, this is kind of a difficult procedure. So at the left hand side there is a picture of one of these little pads. If you open up the plastic around the instrument here, which is only maybe 2.5, 3 inches long. If you open up the casing, there's a pad that looks like this. So over on the left is where you put your sample. In this case it's blood, but it could be sputum, a swab on the inside of the mouth, or the nose, whatever the best sample is. And there are pads of paper and filter paper all along this little strip. The sample is going to move from left to right towards the absorbent pad. And at different places here, there are antibodies. So on the gray pad, there is the first set of antibody attached to some sort of other material, like maybe gold or some other chemical. And as the sample is going to move from left to right through this conjugate pad, if the person has the particular antibody, let's say to HIV, it attaches to the materials that are on the conjugate pad. Then it keeps flowing to the right, and it hits that test line, and there's another antibody on that test line, and now you get another attachment, and then it keeps flowing to the right to the control line, and then finally over to the absorbent pad. So you've got a couple of different antibodies that are on this test strip that will be used to look for the particular thing, either antibody or antigen, that is in the patient. After a period of time, only maybe a couple of minutes, you look for lines, visible lines, pink lines, blue lines on the test strip. There's a very good video. Actually, there are many pretty good animations of this online. So as I said, this is quick. It is very easy. These are cheap to make. If you make them by the millions, they turn out to be something like about $1 each to make. Other ways to identify microorganisms: PCR, also done for the novel coronavirus, the SARS-CoV-2. And you can use gene probes, you can do DNA sequencing. So these are more molecular genetics testing. Let me stop here, and in the next video I'm going to talk about vaccinations and then